Hello, we're going to continue our unit on DNA and talk about biotechnology. We talked a little bit about it when we talked about restriction enzymes and the gel electrophoresis, but now we're going to learn a little bit more terminology. Biotechnology is the use of organisms or their components to make useful products. Most of them depend on the bacteria E. coli. Genetic engineering is the direct manipulation of genes for practical purposes. An organism's genotype can be modified according to human design by introducing genes that have never been present in the chromosome or that species. So it's a pretty cool concept. We do have three main types of cloning, and if you think of cloning as an exact copy, that will help you in understanding this. All right. Pretty much it does not increase genetic diversity. So cloning makes an exact copy. Okay, it is helpful because they have been able to clone rare animals like the guan, which is the Asian ox. They cloned a frog in 1950. 1997, they cloned Dolly the sheep. They've been able to clone horses, cats, cows, goats, and so forth. The first type of cloning is called nuclear transplantation. This is pretty much animal cloning. This is going to replace the nucleus of an egg cell or a zygote with a nucleus of an adult somatic cell. It will have the cell divide, creating a blastocyst, which is a big ball of cells. All right. Reproductive cloning, if you have a mammal, okay, well, what do you need to do further? Okay, so the blastocyst is implanted into the uterus for further development. So the Dolly was given birth by its mom, all right? So it was implanted in its mom, and then Dolly was born. Okay. Therapeutic cloning is producing the embryonic stem cells for therapeutic treatment. Now you get the stem cells from the embryo. And what's so neat about the stem cells is they can give rise to all different types of body cells. So again, cloning is making an exact copy. There's no genetic diversity, and they have been doing it since the 50s. Recombinant DNA is combining genes from different sources of a DNA molecule. And you can use different species using plasmids in particular. If you remember, plasmids were circular pieces of DNA. So molecules that can contain DNA sequence derived from different sources that have been joined together in a laboratory is what recombinant DNA is. Insulin was the first usable human protein to be produced in bacterial cells, and it's pretty good. There's a other thing that is used in the ER to dissolve blood clots that block coronary arteries, causing heart attacks. That's called TPA. There's a thing called frostband that you could place on plants that it won't allow the frost to adhere. So this whole recombinant DNA has been pretty impressive. And also it can create what are called transgenic animals, which are animals that produce genes from different species. Okay, so with this, let's talk about how we would clone the gene or so forth. First of all, we would take the plasmid and we would isolate it. Again, the plasmid is a circular piece of DNA. Okay, it will then insert that gene of interest from the other cell and combine the two. The bacteria will then reproduce it, forming a clone of cells. So gene cloning is the production of multiple identical copies of a gene carrying a piece of DNA, or the recombinant DNA that we have. Okay, and this is just showing you the whole process. So recombinant DNA is DNA from two different sources combining together. And these are just some products of recombinant DNA. Okay, PCR is another very cool technology advancement. It stands for polymerase chain reaction. It was developed in 1985, and pretty much it copies DNA or amplifies the DNA. It's a cycle of heating and cooling, and that could generate up to 100 billion copies of DNA in 4 hours. And what they have actually done, they've taken a woolly mammoth that was frozen about 40,000 years ago, and it has amplified the DNA. So it's pretty much a copy machine of DNA. It's a process of heating and cooling that will make multiple copies. And you can see you start off with one copy, then you have two, four, six, no, 2, 4, 8, not 6, 8, 2, 4, 8, 16, 32, and so forth. Okay, so genetically modified organisms have acquired genes by artificial means, by that use of plasmids. So it's very little bit more sophisticated with the recombinant DNA because we're making a transgenic organism or a animal that possesses genes from different species. Okay, and it is possible for plants all the way through animals. Pretty much the desired trait is inserted into a plasmid, then the plasmid is placed into the organism, and then the organism reproduces it. A lot of our crops are genetically modified. We call them GMO. It makes the plants resistant to herbicides or pests. Golden rice is a transgenic variety with daffodil genes. So it produces grains that contain vitamin A to help with malnourished countries. If you have any questions at all, please see me tomorrow. Thanks.